Good evening, one and all present here. Myself, Dr. Pranita Gulhane, on the occasion of 124th birth anniversary of Dr. Panjabrao alias Bhausaheb Deshmukh, founder president of Shri Shivaji Education Society, Amravati, Science College, Congress Nagar, Nagpur, in collaboration with Microbiologist Society India, organizing a lecture series based on the theme Indians Speaking from Abroad. So on behalf of the organizing committees, Honorable Principal Professor Mahendra Dhore sir, Principal Science College, Congress Nagar, Nagpur, and Honorable President, respected Dr. A.M. Deshmukh sir, President of Microbiologist Society India, on behalf of the organizing committee, I welcome all the participants of today's lecture as well as our eminent guest speaker, Dr. Sajad Khan sir. His topic of the today's lecture is biosurfactants, its application in medicine as antiviral agent. So, firstly, I take the opportunity to welcome and to introduce our guest speaker of today's guest lecture, Dr. Mohammad Sajad Ahmed Khan sir. He is currently working as an assistant professor in biology at Imam Abdul Rahman bin Faisal University, Dammam, Kingdom of Saudi Arabia. He has pursued his PhD in 2012 from Aligarh Muslim University, Aligarh, India. He has his work experience as a postdoctoral research as well as teaching experience of more than 10 years, as well as pre-doctoral research and teaching experience more than 4 years. He pursued his BSc as well as MSc from Aligarh Muslim University, Aligarh. He has postdoctoral research experience from 2012 to 13 at CSIR Institute of Microbial Technology, Chandigarh. Then from 2013 to 14 at Department of Paraclinical Sciences, University of Pretoria, South Africa. Then at Biotechnology Unit, School of Chemical Engineering, South Korea, as well as Department of Basic Sciences from Saudi Arabia. In his pre-doctoral research experience, he was junior research assistant at Council of Science and Technology, Uttar Pradesh from India, as well as postgraduate teacher in biology from Fatima Girls College, India, as well as lecturer in microbiology and senior research fellow in Indian Council of Medical Research, Aligarh Muslim University, Aligarh, India. He has currently involved in studying the bioactivities of drugs as well as nanoparticles and natural compounds for their antifungal, antibacterial, and anti-tumor properties. He is also investigating the production and chemical characterization of biosurfactants produced by yeast and its application. He is studying on the problem of drug resistance and bacterial biofilm formation of the hospital wastewater, fecal and domestic sanitation, and its removal using biological agents. He is also exploring the interaction of host immune cells and pathogens and the effects of drugs and compounds on their interaction during the pathogenesis. So he has his expertise in general bacteriology, mycology, virology, and molecular biology, as well as clinical microbiology and immunology, microbial ecology, industrial microbiology, applied microbiology, and nanotechnology and human biology. In his awards and merits, he is the recipient of many awards and accolades, as well as he has chaired the various sessions as an invited guest speaker, as well as co-chairman in different national and international conferences, as well as he has received MSc Gold Medal in 2003, as well as Postgraduate Merit Scholarship in 2003. He has also received J.B. Bhatt Memorial Award in 2011 from Association of Microbiologists of India, as well as he has received Outstanding Scientist Award by Agricultural and Environmental Technology Development Society India. He has also received Best Poster Presentation Award in 2011 from Saudi International Pharmaceutical Sciences Meetings and Workshop, which has been carried out in 2021 in Saudi Arabia. He has taken membership of various societies and associations, which includes Microbiologist Society India, as well as Saudi Scientific Society for Biomedical Engineering, International Society for Infectious Diseases, Association of Microbiologists of India, Indian Science Congress Association, as well as American Society for Microbiology. He has carried out various research projects funded by different funding agencies, as well as the editor of various journals and reviewer of the reputed journals. He has many research publications to his credit in national and international repute of journals, as well as he has published two books which are published in Elsewhere, Netherlands, as well as Saarbrücken, Germany. He has published more than 15 research chapters in different prestigious publications of the books, as well as he has presented many national and international uh, many research papers in national and international conferences proceedings. So thank you very much, sir, for accepting our invitation as a guest speaker in today's guest lecture. So I must and I am very much thankful to you and requesting you to please share your presentation, please share your information on the topic biosurfactants application in medicine as antiviral agent. Please.
Okay. Thank you, Dr. Pranita, for introducing me at this platform, but it was quite lengthy introduction. Anyway, it's okay. And I'm just working here in the MAM University since 2015 as an assistant professor. And it's my privilege to be the part of MBSI. And I would like to thank the president, Dr. A.M. Deshmukh sir, for giving me an opportunity to present a piece of my work at this platform. Actually, my expertise is on the natural products, especially for the essential oils and the plant products, exploring their antimicrobial activities, especially against the drug-resistant Candida albicans. And biosurfactants, of course, but this topic that I have chosen for the today's presentation is like general information on the biosurfactants and its application in medicine, especially targeting on the antiviral mechanism of action, how the biosurfactant is going to be like a good candidate as a antiviral agent. So just let me share my screen. Is it visible to all?
Yes, sir, it is visible.
Yes. So the topic is the biosurfactants, where we are going to see how this biosurfactants they can be used as a very potent antiviral agent. So the background of this study or the article is basically focused on the recently we had the coronavirus disease for the last 2-3 years, and since the outbreak of this COVID-19, it has threatened public health at the large scale. It killed vulnerable persons, and the death rate was very high. And at that time, there was no available medicines specifically for the coronavirus. We have antiviral drugs, but they were not effective against the coronavirus. So this work is especially will focus on to highlight biosurfactant as an ideal candidate to be explored as an antiviral drug against many viruses, especially coronaviruses. So in general, for the very brief information, as we know that viruses, they are of different shape and size, like helical viruses, polyhedral viruses, spherical and complex viruses. All of these viruses, they have the genome, which is surrounded by the protein capsid. And sometimes they are enveloped viruses and sometimes they are non-enveloped. So regarding the coronavirus, if we classify the coronavirus, we see just a moment. So it could be like DNA viruses or the RNA viruses, and based on the genome that the viruses have. And here the coronavirus, it belongs to the family Coronaviridae, that belongs to the single strand RNA. And so if you compare this coronavirus in short with the other viruses, like a comparison, if we see the coronavirus compared to the influenza virus and the HIV virus, these all are RNA genome viruses. But here, the important thing is that influenza virus and this HIV virus, they do not contain the proofreading enzymes, but the coronavirus has proofreading enzymes that makes it something unique compared to these two viruses. So the problem is that once we had an outbreak of coronavirus, the COVID-19, in the previous years, we had the SARS-CoV and MERS-CoV. So against these two diseases, we had antiviral drugs like ribavirin and lopinavir. And these drugs, they are supposed to be good antiviral agent, but they were incapable of eradicating the COVID-19 virus. Although one of the antiviral drug remdesivir, it is very good and active against these viruses. It's supposed to be a very ideal antiviral agent against RNA viruses, but in case of COVID-19, it's not very effective. On the other hand, we have some drug like antimalarial drug like chloroquine, and also some antiprotozoal agent like nitazoxanide. These two drugs, they are considered as a drug repurposing agent, and these are also considered as the antiviral drug, but still they have antiviral potential, but still they are under clinical trial, and sometimes their use is found to be controversial. So considering this, the available antiviral agents, they have different mechanism of actions, like some are targeting the translation for the virus, and some are targeting replication, transcription, and like chloroquine and hydroxychloroquine, they block the endocytosis. So these are the probable mechanism of action shown by the antiviral drugs. And especially if we consider remdesivir, this drug is supposed to be like acting as a nucleotide analog, where it is acting as an analog of adenine, and it blocks the viral replication. But here comes the role of the proofread enzymes by the coronavirus that has exoribonuclease, and that make it overcome this mechanism of action. So the remdesivir is not able to be actively killing the coronavirus. On the other hand, this nitazoxanide, this drug is supposed to be a good antiviral agent, but still the research is going on and they are not approved for use against the coronaviruses. So considering these factors, there is immediate need to find out some drugs which are especially active against the coronaviruses, and which can behave as or act as immunomodulating agents or certain vaccines, or like coronaviruses and the other viruses, we have to stop the transmission of the viruses. So solution is that scientists, they are looking for the new antiviral agents that could show the broad spectrum approach. And at this, I would like to show one of the video here that will let you understand the problem that we are facing with the coronavirus and how we can get out of this. Just a moment, let me share my video.
Two or three other people. On average,
Audible?
they would spread it to 2 or 3 other people.
Or audible.
Those
Yeah, okay.
2 or 3 other people would on average spread it to another 2 or 3. And so on, and so on. 1 out of every 200 people who became infected died.
Actually, there is something on the screen
In 1
like
year.
Norton antivirus or like that, sir.
Okay.
Yes, I'm also getting the same. Yeah.
Okay. Is it gone now?
No, sir. It is just on the screen.
Okay.
The Black Friday is there.
Okay.
Something there is.
Black Friday, learn more about it. Norton
Yes,
virus.
yes. Norton virus.
Yeah.
We try to remove that. Yes.
This virus killed over 2 million people worldwide. Now, let's back up.
Something other.
Imagine if the virus were even more contagious. If every infected
It's okay now?
Yes.
person passed it on to an average of 4 or 6 people. And if its fatality rate were much, much higher, closer to 3 in 10. This devastating scenario is smallpox. Smallpox claimed hundreds of millions of lives in the 20th century alone, and plagued humanity for centuries before that. And then, it was gone. We completely wiped it out of existence. So, how did we do it? And can we do it again? It starts with a vaccine. In the late 1700s, a scientist named Edward Jenner developed a brand new way to fight smallpox. He called it vaccination. Before this point, the only way to gain immunity from smallpox was to catch it, either through misfortune or on purpose. Jenner's method was far less dangerous. Word of the vaccine spread quickly. Within 5 years, it was in the Americas. Within 10, it was brought to Asia. Vaccination alone would eventually be enough to drastically reduce cases. And that's partially because of how smallpox is spread. Unlike a disease like malaria, which can spread to humans from mosquitoes, or Ebola, which can spread to humans from bats and other animals, smallpox could only be spread to humans from other humans. There were no animal vectors, which means vaccinating people would slowly eliminate the only way it could spread. By the early 1900s, most of Europe, the United States, and Canada had nearly wiped out smallpox. But countries with fewer resources, civil unrest, or higher population density struggled to effectively vaccinate and contain the virus. And as long as smallpox existed somewhere, it was a threat to people everywhere. True eradication would require a global effort.
Within the framework of the United Nations, a new organization exists to promote the welfare of all people, the World Health Organization.
In 1967, the World Health Organization established a unified plan to eradicate smallpox once and for all. Having a centralized agency changed the game for public health. Nations pulled together and vaccination campaigns spread around the globe. Initially, the goal was simple: vaccinate everyone. But that proved harder in densely populated places like India, where it was hard to track outbreaks and the virus could spread quickly. In 1974, over 15,000 people in India died of smallpox in a span of 5 months. So the World Health Organization proposed a more targeted approach. Instead of trying to vaccinate everyone at once, public health officials focused on infected individuals, using a form of contact tracing. Doctors and volunteers would isolate the person and vaccinate anyone who had come in contact with them. And then, whoever had come in contact with those people, effectively creating a buffer of immunity between the infection and the rest of society. This was called ring vaccination. It worked really well, not just in India, but around the world. And on May 8, 1980, the World Health Organization declared smallpox eradicated. These are the 4 factors that helped us eradicate smallpox. While there's no single path to disease eradication, if one or more of these can't be met, it will make it much harder, if not impossible. So let's talk about COVID-19. Vaccines aren't the problem. New technologies have brought us effective and safe vaccines in record time. The issue with COVID-19 is the way it spreads. Let's start with animal vectors. Researchers believe the SARS-CoV-2 virus originated in bats, which means there's a way for it to spread from animals to humans. So even if we were to completely remove it from the human population, it could reappear just as it did the first time. But the way it spreads from human to human is also a problem. A person with smallpox could only spread the disease if they were showing symptoms. But a person with COVID-19 can be contagious days before that point. Some people never develop symptoms at all. It's much harder to effectively trace something you can't see, or isolate a person that doesn't even realize that they're sick. And then there's this. From the beginning of the pandemic, many countries have taken a nationalist approach to it. Some countries banned exports of protective gear, some countries restricted the export of vital drugs, and the United States pulled funding from the World Health Organization. In a September meeting, the UN Secretary General said, "The pandemic is a clear test of international cooperation, a test that we have essentially failed." Even as vaccines become available, richer countries are buying up the supply, leaving poorer nations behind. And that's a big problem, because leaving the virus to spread unchecked in these other places gives it more time to spread, which increases the chance it will mutate. Variants of the virus are already popping up, and that leaves the whole world at risk. All in all, COVID-19 is not a good candidate for eradication. But eradication isn't the only option. Smallpox is the only human disease we've ever eradicated. It's much more common for us to contain and control disease. And that's where many researchers think we'll land with COVID-19. We'll vaccinate and manage. Lockdowns and quarantines will end. And we might end up with a virus that we manage with an annual vaccine, like the flu. Or something even more mild. As adults and vulnerable populations build immunity, it's possible COVID-19 will become endemic, always around but rarely developing into anything more than a common cold. It's not the best answer, but it's a reality we'll learn to be comfortable with. We'll be able to go back to the way things were before. But we shouldn't. Eradicating smallpox took a colossal effort, centuries to create effective vaccines, and then decades more to build the framework needed for global campaigns. It's a reminder of what we're capable of as a society. But it's also a warning. COVID-19 is far from the worst disease nature has to offer, and this pandemic has shown us that we're not ready for something worse. Because our failure isn't that we can't eradicate COVID-19. It's that we let it rise to pandemic proportions to begin with.
Okay, so
These people are in line to vote.
with this, we understand that the just vaccination, we need vaccination to eradicate this COVID-19, but also it needs a global effort. Means internationally, the collaboration between the countries to take care of this disease, and especially there is a need for finding out antiviral drug. So considering this, we have a hope with this biosurfactant molecules. Is it visible my screen now?
It is visible, sir.
Okay. So in this regard, the biosurfactants, which are secondary metabolites mainly produced by the microorganisms, which could be some bacteria or fungus. And these biosurfactants, they are amphoteric compounds that possess surface and emulsifying activities. They are found to be non-toxic. They are highly biodegradable. And also the low production cost is associated with the production of this biosurfactants at the large scale. So biosurfactants molecules, they have the chemical structures ranging from they could be like glycolipids, the lipopeptides, phospholipids, the neutral lipids, the fatty acids, polysaccharide protein complexes. And the research has shown that the biosurfactants, they possess antibacterial, antifungal, and antiviral activities. So considering these facts, the use of the biosurfactants in the medicine This could be a very good or appropriate therapeutic agents in combating coronaviruses and other such enveloped viruses. So as far as coronavirus is concerned, this virus belongs to the family Coronaviridae, to which already we have two viruses, SARS-CoV and MERS-CoV. In the same family, the coronavirus belongs to. And regarding structure of coronavirus, it has the RNA genome surrounded by the protein capsid and also the lipid membrane, the envelope. So this virus looks simple, but once it is internalized in the animal cell, it is very dangerous and very harmful. It hijacks the host cellular machinery, and it can spread in the cells very fast, causing damage to at large scale inside the body. So to protect the viruses from infection, we have the biosurfactants as an antiviral agent. And regarding the biosurfactants, very brief information like biosurfactants, they classified as a low molecular weight and high molecular weight chemical compounds, which are produced by a lots of bacteria and fungi. And these biosurfactants, they have application in various sectors like in the agriculture, in pharmaceutical, in industries, environmental aspect, and the heavy metal removal. Like in the petrochemical industries, they are used for cleaning the contaminated land and the ocean, liberating oils from the soil. They are also used as a cleaning products in the food industry, like stabilizing agents and emulsifier, in the cosmetic industries, in the textile industries. But we are concerned here with the medical application of biosurfactants. It has antifungal, antibacterial, anti-cancer properties, and also the antiviral properties. If we talk about the antibacterial properties, the biosurfactants have been shown to possess anti-adhesion activity. That is, many of the medical devices, they have been coated by the biosurfactants, different kinds of different biosurfactant molecules, and that prevent the biofilm formation over the devices. And also it is going to kill or disperse the biofilm forming cells. And as an antibacterial agent, it can disrupt the membrane stability of the bacteria and thereby showing the antibiotic activity. So in case of coronavirus, it has got the spikes, the protein, and that makes it envelope quite stable. And it also helps to internalize through the ACE2 receptors over the mammalian cell surface. But here, once the coronavirus is inside, it leads to a kind of a cytokine storm, and also because of inflammation is too much, it's going to damage the cells in the body. And also it once the ACE2 receptors, they are blocked, it results into the negative effects, that is the vasoconstrictions and increased blood pressure in the body. These are the symptoms which are associated with the infection of the coronavirus. But here comes the role of biosurfactant molecules. The biosurfactants, they are amphipathic, means they have both hydrophobic as well as hydrophilic ends. So they can interact with the hydrophobic domain. They can interact with the lipid layer of the virus, and thereby they can on the other hand, they can interact with the water molecule, and thereby they can destabilize the membrane structure of the virus and leading to the killing of the viruses. So in addition to that, the biosurfactant, they have property to form micelle structure, and once they form the micelle structure, it enhances their utility as a directly targeting the virus, and also it's quite useful as a liposome or the drug delivery. And this could be a very important aspect of this biosurfactant that can be used in the discovery of antiviral drugs. And considering the antiviral application of biosurfactants, as the research shows already, that the biosurfactant cyclosporin A, which is produced from the fungus Tolipocaladium inflatum, it is found to inhibit the propagation of the influenza virus. On the other hand, lipopeptide, It's used as adjuvant to stimulate the immune system. Some synthetic lipopeptides, they are used as a vaccine. And sophorolipids, they are found to be good immunomodulators and anti-inflammatory potential. They also have shown. On the other hand, biosurfactants, they are also found to be immunomodulators in case of COVID-19 patients because they show elevated level of cytokines. So means they show very strong inflammatory response, and this cytokine storm is going to exaggerate and kill the healthy cells in the COVID-19 patient. So in addition to that, the IL-6 and lymphocytes, they also leads to the increased pulmonary damages. So these are the symptoms which are found in the COVID-19 patients. And it has been reported that different class of biosurfactants like glycolipids and cyclic lipopeptides like surfactant, they have anti-inflammatory potential because they have the ability to produce cationic proteins. They also suppress the reactive oxygen species and lysozyme potential. Like, for example, once the coronavirus enters in the cells, it leads to the increased cytokine storm, but the biosurfactant molecule, they reduces the ROS stress and the cationic production. It may inhibit the, it can reduce the elevated level of cytokine production. And that is a good for, as an ideal candidate to treat the coronavirus infection. On the other hand, the biosurfactants, they can also be used against the acute respiratory distress syndrome, ARDS. And this is one of the symptoms which is shown by the COVID-19 patient, characterized by the buildup of the fluid in the patient's alveoli. And once the alveoli is filled with the fluid, it results into the inefficient oxygen transfer and resulting into the hypoxia. So one of the leading cause for this syndrome is that is the surfactant dysfunction that happens because of the attack of the SARS-CoV virus. So as we see inside the alveoli, there is a surfactant layer which is partition between the air and the alveolar fluid, and this is responsible for maintaining the proper shape of the alveoli. But once the COVID-19 virus attack the lung cells, this surfactant layer is destroyed. As a result, the alveoli, they are filled up with the fluids, and that results into the hypoxia. So once the hypoxia is there, the patient needs mainly the ventilator support because they have no enough oxygen supply to their lungs. So, but here comes the problem is that is socioeconomic factors because the ventilators, like especially in the developing countries like India, we do not have such a huge infrastructure where we can support the high level of ventilators because it is associated with the cost, space, and training the people to run them. So to overcome this process, to overcome all these factors, the biosurfactants could be considered as the ideal candidate because the biosurfactants, they are amphipathic molecule, they show low biotoxicity, and they can be used as a direct treatment for ARDS through solubilizing the alveolar substrate. So on this way, they can overcome this socioeconomic barrier of cost, space, and training for the ventilators. It can be avoided if we use biosurfactant as a drug candidate. The research is going on, and we may have some future antivirus drug based on the biosurfactant against the COVID-19. And biosurfactants and drug delivery is one of the application of biosurfactants in a drug delivery because for the COVID-19, we need such a drug that should not be compromised during its structure should not be compromised during the delivery process. So it should be able to be delivered at the target site of the interest. So SARS-CoV here, because SARS-CoV, it mainly attacked the lungs. So the biosurfactant molecules, they can be easily given in the form of aerosol and lozenge that can be directly reaching to the lung tissues and acting against the virus because it has both cationic and anionic properties. In addition to that, biosurfactants, they show a very good solubility, and that makes them an ideal candidate for the administration and a drug for the at the target site. So not only the biosurfactant molecule, they provide safe passes for the drug to the target. In addition to that, they have twofold activity, like they act as a delivery agent, and they also acting as a natural antiviral properties, and they can also relieve the surfactant dysfunction in the alveoli. So they can be used to treat the symptoms of ARDS. In addition to that, they can also be useful in the vaccine development, where you can see nowadays we have mRNA-based vaccine against the coronavirus. So here the mRNA, which is against the spike protein, is inserted in the patient, but we cannot insert the mRNA directly into the cells because it can be deactivated by the ribonucleases. So we need some nanoparticle coating of biosurfactant molecule to stabilize the mRNA and deliver to the target site, and then the antibodies will be produced. So this biosurfactant also has a role in drug delivery, plus it also an ideal candidate for the development of the vaccine. In addition to that, biosurfactants, they are also used as cleaning products. Like we know very well that in the hand wash, they are used very commonly. So treating not only by the medicine against the virus, we also need social distancing, and so that the spread of the virus is stopped. So in this case, the biosurfactants, they are also, again, because they have amphipathic nature, they are very good candidate, and they are already used in the cleaning products like the soap and detergent. So like we see many of the cleaning solutions and the detergent, they all have biosurfactants, and they are very good they are very good antiviral compounds. So one question is that as we have practiced by washing our hand using the soap or sanitizer, but we compare, if you make a comparison between the sanitizer and the soap is far superior in use compared to the sanitizer. Although sanitizer is also killing the viruses, but it cannot remove the dirt of the dead particle of the viruses out of your hand. But when you are using a soap, in the soap, there are biosurfactant molecules, and biosurfactant molecules, they can disturb or destabilize the lipid layer of the virus. And also, because of the micelle formation, it can take away the dead particles and wash out of your hand. So it is much better to use soap compared to the sanitizer. So implication of biosurfactant is that the research is gaining interest as a biosurfactant to develop as an antiviral agent. Some of the biosurfactants like cellubiose, rhamnose, and trehalose, they have shown the very excellent surface active properties like emulsifying activity, dispersing activity, solubilizing activity, foaming, penetrating, and wetting actions. Altogether, these are the properties of the biosurfactants molecules that can be utilized to overall make biosurfactant as a very ideal therapeutic candidate against the coronaviruses and other related viruses as well. So we can say that biosurfactants can be used to treat the symptoms of the coronavirus. It can also target the virus infected cell. It can also be used in a cleaning agents, preventing the spread of corona. So it's like a multi-purpose drug candidate that can deal with the symptoms of the COVID-19. It can also kill the virus infected cell, cleaning of the virus infected cell, and also the cleaning agent to prevent the spread of coronavirus. So in the future, perspective, we can use biosurfactants molecules or different class of biosurfactants compound. They have been already in use in pharmacological and food industries. For example, we already have used biosurfactants in the gummies. So we can make such kind of gummies, the biosurfactant molecules, which are very good antiviral compound. So that can be used to provide the symptomatic relief against the patient directly, because when we chew the gummies, it will directly go to the lungs. And and also nowadays there are some antiviral face masks can be used up. So overall, this biosurfactant has a very good future to be considered as an antiviral drug. And secondly, the microorganisms, they synthesize this biosurfactant molecules, and we can easily yield or scale up the yield of this biosurfactant at a very low cost. So of course, this biosurfactant is going to be a very good for the drug industries to develop at the large scale. So in overall, we found that biosurfactants can be used to prevent the outbreak of COVID-19 and other related viruses, because it has implication in the therapeutics by directly as a drug, also development of the vaccine and strengthening the immune system by lowering the cytokine storm, and also in the environmental to stop the spread, and also used as a cleaning agents like disinfectants, detergents. So overall, this biosurfactants is considered to be as a very ideal candidate for controlling the outbreak and prevention of coronavirus diseases. So this is all from my side for today's, and thank you.
Yes, sir. Thank you very much for your detailed information regarding biosurfactants and its application. So up till now, just the applications of biosurfactants are in food and cosmetics and all other areas, but in the medicine, specifically as an antiviral agent, it shows great effect. Recently, we have faced many problems regarding coronavirus in its pandemic situation. So it is great, sir. There is one question
Yeah, of course,
in
yes,
my
please.
mind, that is it effective to include or to add biosurfactants in that sanitizers, or it is better to use it in the hand washing soaps, like, means if we compare, because you have given the
Sanitizer, if
applications in both, means sanitizers also and the soaps also. So what is the better one?
better one is soap, because when you are using the soap, you have to wash your hand. But once you apply only sanitizer, you are not going to wash your hand, and the virus, that particle still remain on your skin. But in case of when you are using soap, you have to wash your hands. So because this biosurfactant can interact with the both virus and also with the water, so it can easily remove the entire virus from your hand. Means its efficiency is much better than the form of sanitizers compared to the sanitizer.
Yes, but just I have the query, like in
Pranita,
every soaps or, yes, sir.
if you put a biosurfactant in the sanitizer, it will be denatured. It's a protein molecule, or it is a lipid. If you put in a sanitizer, it will be denatured. So in the soap, as Sadaj is saying, it is better. It's better in the soap.
Okay. So that is good, sir. So that is my query.
Yes, sir.
That in every product, is there the biosurfactant is already added, or what? As the general means, for the general knowledge point of view from my side. If
I will tell you one example.
Yes, sir.
Pranita, I will tell you one example. There is a biosurfactant known as rhamnolipid. Rhamnolipid. Supposing you have a burn on the skin. If you apply an ointment with biosurfactant inside, the wound healing is very fast. Understand? If you put in the ointment biosurfactant and apply to the wound, the wound healing is very fast because biosurfactant is antimicrobial, not only antivirus, but also anti-yeast, antibacteria. So wound healing is very, very fast. I have just submitted a project to ICMR for an emeritus scientist on this. I want to add biosurfactant to the ointment to be applied on burns. And I hope I'll be successful in getting the
Yeah, of course, inshallah.
Okay. Sir, can you please minimize your screen?
Hello. Yeah. Sorry, can you hear me?
Yeah.
Thank you. Thanks, Dr. Sajjad. The last slide you put up, that's our work that we did in UK and published in Frontiers in Microbiology. Thank you for acknowledging our work. Yeah, we have been working nearly 30 years on this topic, biosurfactant, rhamnolipid, sulforolipid, lipopeptide, and some of them moved to industrial scale. So we have tried to produce in a 100-liter fermenter, and also we set up a spin-out company related to this in UK, and currently we are doing biosurfactant platform for antiviral, and it's mainly on I would say it's a in vitro studies. We started to do mainly directly on COVID-19 variants, so we have access to the laboratory. So it's your presentation, it's fantastic. So you have updated very nicely. That's really good. Even though I have been doing, I didn't have much time to go through everything. Today, you mainly on the COVID-19 topic, even though we published this paper, afterwards we worked on a laboratory level various things. But it's today's talk is it's an update for me. And nicely you have explained the interactions and how it can be done. So do you have research lab to do any of this work? Because we have collaborators in Saudi Arabia.
We have collaborators in Saudi Arabia. We are directing towards this research. Okay. So we are just showing interest, and we have started just initial work in our IRNC lab here, but not too much. But inshallah, we will go through it in some few years.
Fantastic. Do you have a tensiometer or any equipment which you can start with?
Yes, that's some kind of a challenge process kind of work. So we are collaborating with
if you don't have no worries, we can start it.
We are working on it. Yeah.
Oh, good. Because this topic now we are also looking various partners. For example, King Saud University, we have access, but your institution, I haven't looked. Maybe I'm just looking if 2 or 3 those who knows the topic well, then it would be possible to run that project effectively to get something good out of it. Yeah, that's good.
We can be in touch. Maybe in the future we can have something to the positive outcome.
Yeah. Yes, Dr.
Rahman, I'm very happy to. Dr. Rahman, I'm very happy to see you here.
Oh, thank you. Yeah.
You are authority on biosurfactants, and very soon I'm going to invite you for a special talk on MBSI.
Okay, Professor. Thank you. Yeah,
we'll have a sort of panel discussion with 3 of us: myself, Sajjad, and yourself.
Okay. Yeah, that would be nice. Yeah, I can share some of my experience.
It will be my pleasure.
And
it will be my pleasure.
Thank you. Good to learn from all of you, great scientists.
Thank you. Yeah, it's a very nice start today. It's amazing. And thanks, Professor. And we have referred your paper also, Professor Singh's team. I think for my PhD in 1993, they got a publication with Professor Ibrahim Bennett. Maybe it's good to share some good news now. That time I was doing my master's and PhD. So you have excellent papers, review article on biosurfactant. After reading that article, initially I was doing my PhD on biocatalysts. Then I switched to biosurfactant, And then we have prepared a detailed proposal for CSA or SRF. I got the funding to do for 3 years project. Then I got postdoctoral work with the Professor Ibrahim Bennett. And then I went to US. So probably your article with Ibrahim Bennett would be the start for my research. So it's now we have built up the nearly 30 years.
There's a spark for many researchers in the biosurfactant that article. Yeah.
So thank you, Professor. Yeah, it's all it's amazing journey. Thank you. Yeah, I'd be happy to discuss in detail when we have a panel discussion. It's excellent area. Thank you. Thanks to the organizer, Dr. Pranita. It's excellent. Yeah, thank you.
Yes, sir. Thank you very much, sir, for your appreciation for the said topic as well as our speaker too. So we feel really very great that he has really shared very great information on the biosurfactants and its applications. So if anybody have any query, they can please ask, or we can conclude here.
I just want to ask one question to Sajad. Sajad, even the microbial cell can be considered as a biosurfactant?
Microbial cell as such?
As such, the whole cell.
The whole cell, if you can process it, if they are producer of biosurfactant.
No, actually, the cell wall is composed of a mosaic of lipids and proteins. So the cell wall itself also is a biosurfactant. The cell wall is a biosurfactant naturally. So even the cell wall is classified
the whole cell
as a biosurfactant. The whole cell.
Yes, sir. Okay.
So maybe I can add in that aspect. For example, whole cell and cell-free supernatant, if you consider. If the whole cell means all these surfactant molecules, they have a hydrophobic and a hydrophilic nature. I'm not sure whether you can see the cursor on the screen and on that slide. No, probably you may not be able to see. So you can, in the micelle formation, in that picture, the last picture, what you have put up. So it has the circular, the last one, and then micelle, yes, that one. So this micelle, you can see that circular part and then long chain. So the circular part is the hydrophilic part. So that could be the carbohydrate or lipid, and the long chain fatty acid, that's the hydrophobic part. So, but it is in oil in water or water in oil emulsions we can create. And these structural variation, already you have highlighted some during your presentation. Whereas in bacteria, the whole cell, if you consider, where we will find the water, if it is the whole cell, as a cell, it's a hydrophobic nature, but we need some point to, the hydrophilic to attach to water, then only it will create emulsion. Or else after you leave it, all the cells will go to the top. It's almost like, probably it will not create that emulsion type of approach, what we need for micelle formation. For micelle formation, we need both hydrophobic and hydrophilic. So probably, in my knowledge, I think the structural, the whole cell, if we use that, either it could be only one part because of the cellular structure. But it's just a straightforward answer I'm saying, but maybe if we dig in depth, we may get some other insight. That's we can try. But any extracellular lipids present or produced by these organisms, that will be dispersed in the liquid. So that's for example, as Professor Singh says, if whole cell means the simple one, yogurt or lactobacillus, if the lactobacillus can produce biosurfactant, then the whole cell, if we take it and then mix it, can we make it as soap? Is it possible? We need to think about it. But if the surfactant produced in that liquid, definitely it is possible with that mix, because there are some surfactant molecules there. You'll be able to create that. Yeah, that's kind of so. But it's interesting to explore. Thank you. Thanks, Professor.
We
You
have tablets.
trigger my
We have
thought process
tablets.
now. Thank you.
We have tablets of lactobacillus known as Sporlac. They're available readily anywhere in India. So if your stomach is bad, you take lactobacillus as a whole cell in your stomach.
Yes.
It multiplies rapidly in the stomach, the whole cell, and you're cured of the dysentery. So the whole cell is a biosurfactant.
Sorry, maybe we will discuss it separately. The surfactant, it has its own function. It needs a hydrophobic and hydrophilic moieties. Then only we can classify that as a surfactant. Whole cell may not have that option. Where is the hydrophilic part or hydrophobic, whatever it is? Yeah. I'm sorry. Sorry, I couldn't hear.
Yes, the sound is not clear.
Yeah.
Not audible.
They are both there. Hydrophobic, and hydrophilic. No, the cell hydrophobic and hydrophilic moieties of both hydrophobic and hydrophilic muties. And that is how the lactobacillus works in the stomach, the whole cell. So the cell wall is already classified as a biosurfactant. Of course, you can have it out in the medium. You can have it out. You can have lipopeptide, you can have rhamnolipid, you can have surfactant in the broth. But a whole cell also is termed as a biosurfactant.
Maybe I will contradict with that. It is impossible for me to accept it because of the whole cell will not have both hydrophobic and hydrophilic moieties at the same time. Maybe I have to look in some
you have to brush up
strong evidence. Yes, yeah.
You have to brush up your knowledge.
Insight on this.
The cell wall
is
composed of hydrophobic and hydrophilic moieties always.
So that okay. So if in that case, then how to create emulsion? Maybe we will discuss when we look at the structures, and then it might be possible.
We can discuss separately.
Separately, yes.
I am concluding that lactobacillus whole cell is a biosurfactant. It works very well in a tummy disorder. It multiplies rapidly in the stomach, and the whole cell actually is a biosurfactant. And we'll discuss later on some other day.
Yeah, that's great. Thank you. Thank you, Professor.
That's great.
All right.
Thank you.
Yes. It is really very nice discussions, so that we the juniors should get updated with the knowledge regarding such
from the great researchers. Yes.
It is an opportunity to us to listen you all. So with the permission of Dr. Deshmukh sir, I take this opportunity to conclude our today's guest lecture. So thank you very much, sir, Dr. Sajad Khan sir, that you have accepted our invitation. So on behalf of the organizing committee, Honorable Principal Professor Mahendra Dhore sir from Science College, Congress Nagar, Nagpur, as well as Honorable President, Microbiology Society India, Dr. A.M. Deshmukh sir, on behalf of the organizing committee, I am very much thankful to you, sir, for your precious lecture. That would be really very great for us. It is a kind request to you that if you could put your email ID in the chat box, then it would be great to our participants as well. This lecture is also live streamed on the YouTube, so that the link will be shared on every group of Microbiology Society as well as our institution. So once again, thank you very much, sir. Thank you.
Yeah, okay, thanks. Thank you.
Thank you.
It's my pleasure to be the part of MBSI and discussing my work over here. Thanks for MBSI, and I'm seeing a part of MBSI is ever increasing. Every day I see the new contacts have been added up, and it's a very good platform for the researchers to come together and share their views, and many activities like conferences, workshops. It's very good, wonderful. So all the
Yes,
best
sir.
for the NBSI for its successful, like AM Deshmukh sir and the Rajneesh sir and many more coordinators are there. So they should take you to the new high for the NBSI in India. That's great.
Thank you very much, sir.
Thank you.
for your best wishes. Thank you.
Yeah.
Thank you,
Thank you.
Pranita.
Bye now.
Thank you.
Yes, sir. Thank you.